Welcome back to BE544. In lectures 1 through 4, we built a strong foundation from histopathology fundamentals and digital slide acquisition to hands-on experience with QPath and ImageScope. Today, we shift gears toward the computational core of our discipline. First, let's clarify the hierarchy terms that underpin this lecture and future ones as well. The first term is artificial intelligence, and this is the broadest concept. It refers to any technique that enables machine to mimic human cognitive functions such as reasoning, perception, and decision-making. There is a sub-branch in it called machine learning, and it involves algorithms that learn patterns from data without being explicitly programmed for every rule. Instead of hard-coded logic, machine learning models infer relationships through statistical optimization. Another sub-branch is called deep learning, which is a specialized branch of machine learning that uses artificial neural networks to automatically extract the hierarchical features from raw data, such as pixels in an image. In digital histopathology, we primarily utilize deep learning because it excels at learning complex visual patterns directly from the whole slide images. Also, eliminating the need for manual feature engineering. So we have machine learning and deep learning, which are branches in the artificial intelligence, and the deep learning specifically is a sub-branch in machine learning. And the aim of machine learning and deep learning is to learn from the data and define the patterns to do our task, such as classification or the discrimination between the different classes or categories. So with this foundation, we can now turn to 4 specific vision tasks these models perform, which are classification, localization, detection, and segmentation. In AI-driven digital pathology, precise terminology is critical, not only for model design, but also for evaluating performance and comparing methodologies. Let us begin with classification. This is the task of assigning a single label or category to an entire image or region. For example, classifying a tissue patch as tumor or non-tumor, or categorizing a breast cancer slide into molecular subtypes such as luminar A or HER2 enriched. And for simplicity, let's assume there is an image here with a cat in it, and the aim of classification is to determine the category of the whole image, so the model can predict the output to be cat, dog, or any other category. Classification operates at the image level and doesn't provide spatial information about where the relevant features are located. It's the simplest form of supervised learning in histopathology, yet foundational for diagnostic screening and triage systems. Next, we introduce localization and detection. Localization answers the question, where is the object of interest? So, I need a location. It typically outputs bounding box coordinates such as left upper corner coordinates, width and height to indicate the position or the location of a lesion. Detection combines classification and localization. It not only identifies what is present, but also where it is, often for multiple objects simultaneously. For instance, a model might locate and label or classify all tumor infiltrating lymphocytes in a field of view. However, detection is limited by its use of rectangular bounding boxes, which cannot capture complex irregular cellular shapes, which is a key limitation in histopathology, where morphological detail is paramount. This brings us to segmentation. And segmentation is the most granular and clinically informative task. Unlike detection, segmentation delineates object boundaries at the pixel level. There are 2 primary types of segmentation. Semantic segmentation, which assigns each pixel a class label such as nucleus, cytoplasm, or stroma, but doesn't distinguish between individual instances of the same class, where all nuclei, for example, appear as one continuous region. If you look here for the semantic segmentation, it segmented all of the sheep as one object, the grass as another object, and the road as a third object. It didn't differentiate between the 3 of them. And here comes instance segmentation. It goes further it differentiates each individual object even if they belong to the same class. So every new class receives a unique identifier, enabling quantitative analysis such as cell counting, nuclear morphology measurements, and the spatial relationships or spatial statistics like nearest neighbor distance. So in this figure, it segmented everything, sheep 1, sheep 2, and sheep 3, and gave each of them a unique ID. Having defined these tasks, let us now examine how they fit into a real-world deep learning pipeline. The first workflow illustrates a tile-based classification approach, which is one of the most common strategies due to the gigapixel size of the whole slide images. The process begins with scanning histopathology slides at high resolution, let's say 100x or 400x. Next, the tumor region is identified either manually by a pathologist or via an automated tissue detector. So let's say this is a tumor region, and here we segmented it to extract only the region of interest, which is the tumor region, and ignore other regions, and then subdivide it into smaller, manageable tiles. For example, tiles with sizes 256 by 256 or 512 by 512 pixels. So as you see here, the authors of this study divided it into tiles as you see the small squares here. These tiles later undergo preprocessing steps, such as normalization to reduce patch effects from staining variability, and data augmentation to improve generalization, and we are going to illustrate those techniques later in the course. But here, the authors applied preprocessing, and generally the aim of preprocessing is to make the data ready for the classification task. Then, a machine learning or deep learning model is then trained on those tiles with associated labels. Let's say tumor and non-tumor, or tumor with grade A, tumor with grade B, and so on. Finally, predictions from individual tiles are aggregated using methods like majority voting or attention-weighted averaging to produce a patient-level diagnosis. So, if this whole slide image reflects a specific patient and then divided into a set of tiles, let's say 1K tiles, extracted from this patient's whole slide image, and used this 1K tiles into the classification, then we will get 1K predictions. So to determine if this patient has a tumor or not, or what's the grade of the tumor depending on our task, we will aggregate all of them. Let's say we will apply majority voting, which is voting on which class has the higher frequency or the higher count, and it will be the output for the whole slide image. So let's say around 950 tiles predicted to be tumor and 50 only predicted to be normal. So the patient, depending on the majority voting, will be tumor. So this hierarchical inference bridges macroscopic detail and clinical decision making. A second example. This example demonstrates multi-scale validation framework applied to cardiac histopathology. Here, the patients are split into training and testing sets, ensuring no data leakage. And data leakage is an important concept in artificial intelligence. And every author or researcher or anyone using machine learning should ensure that there is no data leakage. And the meaning basically of data leakage is you will find a data in the training and the same data in testing. So the leakage here means you find duplicates with the same patient or the same case into training and testing. So simply the meaning of training dataset and testing dataset is a training dataset is used to teach the model, whether it's machine learning or deep learning, to detect the patterns from this dataset or subset. So the model at this case saw the data and aware of it, but the testing dataset or subset will not be seen during the training. It will only seen after the model is trained. It's like a student is learning during the whole course, and at the end, the student will be tested on unseen exam or unseen questions. So if there are some samples from the training are found in the testing, this means there is a leakage, and the consequences of leakage can be danger because the model will be tested on the same data that the model learned on. Is there any problem in this case? For sure, because in this case, the model may say I'm getting a very high performance in the testing, but in real, on any future unseen data, it will perform badly. And the problem in this case will be called overfitting. The model is trained well, but on unseen data or unseen patients, it will perform badly. So the authors ensured that there is no data leakage. What's next? Region of interest or ROIs are extracted from whole slide images, and all of them are from the same patients with the same label, such as heart failure or normal case. And this study, which is here, compared two modeling approaches. One is deep learning versus a classical machine learning pipeline using a set of features. And features here mean a set of handcrafted image descriptors. So the authors compared between both of them. At last, they need to evaluate the model or their approach. So the evaluation in that study occurs at both image level, which is called tile accuracy, and patient level, which is diagnostic sensitivity and specificity. Both highlights best practice, which is robust AI systems must be validated at the clinical endpoint, not just on pixel level metrics. And all of these concepts will be covered during the course. How to judge the system on the pixel level and how to judge the system on the patient level. But here we are showing the importance of including machine learning or deep learning into the pipeline to automate the diagnosis and help the medical staff. A third example, in it, the system showcases a multi-stream architecture for multi-class tissue classification. So here, the authors were trying to distinguish between 6 histological categories. For the input tile, the tiles are processed in parallel through individual pre-trained VGG networks. And those networks are deep learning models. Also, these models utilize a concept named transfer learning from natural image datasets. Again, we are going to cover how to use VGG and what's the meaning behind it later in the course. If you notice, the authors in the input level use tiles from different magnifications, 25x, 100x, 400x. Each got a specific model. Let's say this will be the feature extractor point. So we extracted features from each magnification level, and then they are combined into a layer so that all of the features from the different magnification levels are concatenated together, and then perform the classification to get the output in the output layer. So instead of depending only on a single magnification level, as we saw in the first two examples, here the authors got the benefit of using the different magnification levels. And this is another approach. And which one to select, it depends on the problem and the acquired data. Maybe the acquired data got only one magnification levels, and in that case, you are forced to use just the level. But if you have different magnification levels, it's recommended to utilize all of them, because in high magnification levels, you are getting fine details, and lower magnification level, let's say 10x or 25x, you are getting global details. So combining both of them, you are getting more details locally and globally. So before we proceed to coding demonstrations and how to build the deep learning pipeline, let us address first a foundational tool called OpenSlide, as it will help us later to load the data and acquire different tiles from it. So OpenSlide is an open source C library with Python bindings that enables reading of whole slide images in vendor-specific formats such as SVS. To install OpenSlide for Python, you will need both the system library and Python wrapper. So on Windows, precombined binaries are available on the official website. On macOS, you can use on Pro. On Linux, you can use the package manager. And more details about the installation can be found on the OpenSlide link. Then, in Python, run the command pip install OpenSlide Python. And this library is essential for building custom data loaders and preprocessing pipelines, as it provides efficient memory-safe access to the pyramidal OpenSlide images structures. So, depending on your operating system, you will need to access the terminal and ensure first that you installed Python on your operating system. And Python is the programming language that we are going to use. Then, as mentioned, you need to install the OpenSlide package for Python by using the pip command pip install OpenSlide-Python. It will start the installation, and at the end, ensure that it's installed without any errors. After that, you will need to download the course codes provided on this GitHub repository. Simply, on the provided link, you can download the whole repository from code and then download zip file. Or if you are aware about Git commands, you can clone the repository using this link. So if you downloaded the zip file, you will need to extract it to obtain the inner scripts. So once you extract it, you will find the same files as in the repository. Now we can start working on them. Now you can open the scripts folder using any coding editor. Here I'm using PyCharm, but you can use any application like PyCharm or VS Code or any alternative. If you navigate to the lecture scripts, you will find a set of Python scripts, and on each lecture, the repository will be updated by the new codes. So if you opened the original repository after each lecture, you will find more codes are added in this region. And to download a specific code, you can either download the whole repository again or go to the new code, which is required to be downloaded, and then use the download raw file here. Once it's downloaded, move it into your lecture scripts folder. The aim of providing these codes is that you don't have to code from scratch. You would obtain the steps, how to implement that, and you need to be aware about the steps and how to change the inputs and how to read the output. Also, you don't have to memorize everything. You can write comments above any statement or sentence in the code so that when you return back to the same code, you can remember what is done before. So many of our codes will focus on the BACH dataset, which is a publicly available benchmark for breast cancer histology classification, and it contains 4 classes. Normal, which is the healthy breast tissue; benign, which is a non-cancerous proliferations; carcinoma in situ is the malignant cells confined within the basement membrane; and carcinoma invasive is the malignant cells reaching the basement membrane and infiltrating surrounding stroma, which is the hallmark of invasive carcinoma. By all means, we have 4 categories, and later we need to build a pipeline to classify between and discriminate between the different classes here. And to download the dataset, you can either use this link or that link. There is the first link and there is a second link. Also, there is a research paper for this dataset and the original website. So assume that you need to download a single file, not the whole dataset. So it's recommended to use the Kaggle link, as if you scroll down here, you'll find the main directory with the whole slide images here. So if you navigated to the whole slide images folder, you will find the different slides. So scroll down and download this file by simply pressing on it and click on download link here. Do the same for the XML file. If it didn't download and navigated you to another page, you can simply press Ctrl S or save and save it, but ensure first that this file ends with .xml, and this file contains different annotations for the downloaded whole slide image. Now move these 2 files inside the scripts folder, and it's recommended to create another folder called data and move any data into that folder. So if you open the data, you will find the two files. And just to notice, if you find that the downloaded SCVS file has a postfix extension.zip file, you will need to remove it. Now we have the SCVS file and XML file. So that the type of the file is SCVS file here and XML document here. And if you downloaded the whole dataset, just take a copy of these two files into the data folder here. As in this lecture, we will deal only with this slide. Now, let's return to our editor. We have the lecture scripts with the lectures here, with the scripts here, and the folder data with the two files as here. Now, let's open the first script. So, the aim of this script is to read the whole slide image and get the knowledge of accessing the metadata and the different properties of the whole slide image. So here we imported the OpenSlide library that we installed from a while. Also, for visualization purposes, we are importing the matplotlib package, and you can install it using also the pip command pip install matplotlib. Next, we define the path of the SVS file that we downloaded, and here we set the relative path, which is data/ao2.svs, which is data followed by the file. Then we use the function OpenSlide that accepts the path of the SVS file and returns an object with the slide. Now, using that variable or that object, you can access different metadata and information about the slide, such as the dimensions, the number of resolution levels, the dimensions at each level. And to print them in front of you on the command window, you can use the print command and put here whatever you want to print. Also, this part prints the downsampling factors in each level, and this reflects the magnification levels, such as 1x, 4x, 16x. Here, we printed all of the metadata properties just in case if you don't know how to access a specific one. And at the end of the code, we displayed a figure, and this figure contains an extracted region from the whole slide image, as it's difficult or can be impossible to visualize the whole slide image at, let's say, the highest magnification level on the screen, because the gigapixel size of each. That's why we are extracting tiles or small regions. So to extract a region, you can use the function slide.readRegion, and this function accepts three things. First, the XY coordinates, which will be the top left corner of the region. Which magnification level are you targeting, whether it's 0, 1, or 2, or any magnification level, and the size of the region to be extracted. And here we are extracting 1K by 1K image. This will return a rectangular region, and we used the mshow function from the matplotlib library to display the figure. If you executed the code and found an error like that, which is mostly happening in Windows operating system, you can use the pip command pip install OpenSlide-bin and execute it and try to rerun the code. So once you run the code again, you will find that this is the output of the script, and this is the visualization of the extracted region at level 2 as a specified coordinates. And as you see, we have three levels: level 0 with these dimensions, level 1 with those dimensions, and the same for level 2. And as you see here, the downsamples, which reflects the magnification levels for level 0 is 1, so it's 1x, and in level 1, which is the second level, because in Python we start from index 0, it's 4x, and in level 2, which is the third magnification level, it's 16x. And we have here around 32 different properties. And here you will find different metadata, such as the date of the file, the name, the dimensions, and so on. So the aim of this code is to understand how to use the different properties and how to crop a specific region from the whole slide image and display it. Now let's move to the second code. In this file, we are trying to read the annotations, and the annotations here are stored in XML file. And you can read different annotations file, such as the annotations exported in QPath or ImageScoop. But here, the annotations are predefined by the authors of the dataset. So this code uses a function called extract PACH annotations from XML, and I have coded this function inside the helpers file here. So if you double click on it, you will find the function implementation. And also if you opened the XML file, as you see here, you will need to know the steps of gathering the different annotations, which is the implementation of this function. First, we imported a package called XML, and here we used the class element tree that will deal with this XML file as a tree. So first, we parsed the XML file to be a tree, and a tree because the XML file or the annotation file contains parents and children, and each node or object here may contain properties or attributes and other children. So first here we got the root, and the root in this case is this object. Then we created a annotation list as an empty list in Python. Then we asked the root to find all annotation, and to access a specific node, here we need to know the keyword. So this keyword is annotation. That's why we asked to get all of the annotations inside the root. Then we checked if there are no annotations, return the empty list. Otherwise, continue. So here we have different annotations. Inside each annotation, there are regions, and the regions has a keyword or a child called region. So for each annotation, we try to find the regions with the keyword region. And for each region, as you see here, there is different vertices. So inside each region, I'm trying to find each vertex here. So I use the keyword vertex inside the region. And for each vertex, I need the X value, the Y value to be acquired. So here I accessed the attribute X and the attribute Y to be the coordinates combined together into the coordinates list. Now I have the coordinates, but as mentioned, we have 4 classes. So I need to append also the class for this region. So I accessed an attribute called text from the region. So in the region here, if you go to the right, you will find different attributes, and one of them is called text with a value of benign. And this value differs according to the region. So the class will be stored for this dataset in the text attribute. So to build such a code for any other dataset, you need to follow the same approach. Try to recognize the pattern and loop on the children one by one until you access the nodes for the coordinates alongside the category. That's why I specify this function is to extract the PACH annotations, as it may differ from a dataset to another. So you are supposed to know the flow, what's the input and what's the output. You don't have to rewrite this function from scratch again. So the input for this function is 2 arguments, the XML file and verbose. The XML file is the path for the annotation. Verbose is a flag, whether it's true or false. True to print the number of annotations, the number of regions, the category, and the vertices on the command window. If it's false, this function will not print anything. And this function returns a list, which is the annotation list. And inside this list, there are different elements with the type dictionary. And each one of them contain the coordinates alongside the category. So here we imported the function from the helpers file and called it, passed the argument XML file, which is the path of the XML file that we downloaded earlier, and verbose equals true. So if we executed it, different prints will be printed from the function call, and at the end, the total number of parsed annotations will be printed. So when we executed it, different prints here, we have 17 regions, benign, carcinoma in situ, carcinoma invasive, and we have 17 annotations in this file. Now, in the third code, we need to combine both the whole slide image, which is a slide, and the annotations together, so that we need to draw the annotations on the whole slide image for visualization. And the aim of this step is to validate whether the annotations are correct or not. So here we added two variables, one for the path for the SVS and another for the XML file. Similar to the first code, we created the slide object, and similar to the second code, we extracted the annotations. Then we extracted a thumbnail, and a thumbnail here is a downsampled version from the big whole slide image. So a thumbnail will get the whole slide image in a downsampled version to fit in the memory. So here we acquired a thumbnail with the dimensions of the original dimensions downscaled by 100. So if the original dimensions are around 40K divided by 100, so it will be only 400. The remaining of the code creates a figure. So we displayed the figure and we created an overlay on that figure with the different regions, and the regions got a color depending on the category. So if it's benign, it's colored in green, circinoma and cyto, it's in yellow, invasive in red, and if it's normal, it will be colored in blue. So here I am looping on the annotations, which is output from the function, accessed the x coordinates and y coordinates, and again we are downsampling each coordinate by the same value as here. And if you didn't do that, you are likely adding the coordinates in a different location. Then we created a text with the category name. After that, we filled the polygon and draw that polygon on the thumbnail. So if we executed the code, we will find something like that. As you see, the annotations are colored and there is an overlay text on each of them. For example, the benign is colored in green and has a text of benign. The same for others. Red for invasive, yellow and cyto. And because there is no normal, there is no blue annotations. So let's assume that we need to work on a specific region. Let's say I need to work on the benign region here. So in this case, we need to extract that region specifically in a high resolution manner. Because in thumbnail, if we zoomed in, it has a bad resolution. There is no details. And as mentioned, the thumbnail is just for viewing and validation and ensures that the annotations are correct or the whole slide image is loaded correctly. So what to do to extract this region or any other region at a high resolution? And here comes the fourth code, extract a region. We did the same. We created a slide and obtained the annotation list. For illustration, we are only accessing the first index, which is index 0, or the first region, and you can change that index depending on your target region. And if you are targeting all of the regions and you need to make it automatic, you can wrap this block inside a for loop. But for now, we need to work on the first region with the index 0. Here, I created a function called extract whole slide image region inside the helpers file that accepts the slide and region and returns the region image, the mask, and ROR. The image is the colored output of the region. The mask is an image with colors black and white. White for what's included inside the region and black otherwise. And the ROI is the ending between both of them. And we are going to see what's the meaning visually behind those three variables. But first, let's understand the flow of this function. This function, as mentioned, accepts the slide and target region and returns a tuple of three things: image, mask, and the region of interest. Rest, we validated the inputs to ensure that the data are correct, extracted the XY coordinates of the region, the same for width and height, and ensure that the width and the height are suitable and not below or equal 0. Then we created a shifted coordinates, and we used the shifted coordinates so that the coordinates start from 0 and 0 instead of 20K by 20K, for example. And it's important to create the mask, as we did here. Next, we use the function that we illustrated earlier, which is read region, that accepts the top left corner, the level, which is level 0, which is the highest magnification level, and the width and the height. The width and the height are obtained from here, and those are obtained from the coordinates. So we are trying to find a bounding box that surrounds the target region. To obtain the region of interest, we apply bitwise and bitwise AND simply extracts only what's inside the coordinates or the region. So what's the meaning? Let's execute the code to understand it visually. As you see, this is the image as a rectangle region. This is the mask, and the mask reflects that the white means included and black means excluded. So simply, if you remember, the region was surrounding that portion, which is reflected by the mask. But now I don't need to deal with the whole image or the whole region in this rectangle. I need only the elements or the tiles inside the region, which is categorized as benign, as I don't know what is the category of the outside region. So simply I will apply bitwise AND to obtain the region ROI or region of interest. So the bitwise AND simply multiply each pixel here by 1 or by white or by 255, and anything multiplied by white will be the same thing. But if anything is multiplied by black or 0, it will be 0. So this region is multiplied by black, which is 0 as a value, so the output is black or 0. By doing so, I obtained only this region. Let's say I need the next one. So I will change the index here from 0 to 1. I will execute it, and this is the output. This is the rectangular region, this is the mask, and this is the region of interest. You can do so by any index. We have 17 annotations, so you can access from 0 to 16. Let's say I will access index 12, which is annotation number 13. As you see, the mask bitwise the image, there is a region of interest. So what we did, first, we knew how to read the whole slide image, then how to read the annotations, then how to draw the annotations on whole slide images, and then we can now extract a certain region. It's from the thumbnail, and this is the reflected region of interest. And in another example, there's a thumbnail, there's a region of interest. So, in this lecture, we have illustrated the meaning of artificial intelligence, deep learning, and machine learning, and we went through the 4 tasks in machine learning and deep learning, and then went through the 4 examples or tasks in deep learning or machine learning, which are classification, segmentation, localization, and detection. And we knew them that segmentation has 2 branches, semantic segmentation and instance segmentation. After that, we went through installing the OpenSlide package in Python and recapped the BACH dataset, which contained 4 classes. And after that, we went through the demonstration of 4 scripts from reading the whole slide image at annotation until extracting a specific region at a specific index.